My name is Mike Zyskowski. I'm a Senior Research Program Manager in Microsoft Research, and today I'm introducing the Microsoft Research Sequence Assembler, a sample application that demonstrates the Microsoft Biology Foundation, the .NET Framework, and Microsoft Silverlight for bioinformatics research. The Sequence Assembler is a simple and flexible way to work with genomic data by exposing the features and functions of the Microsoft Biology Foundation, or MBF for short. The Sequence Assembler implements several features of MBF: a set of parsers for common genomic file formats, a set of algorithms for assembly and alignment of genomic data, a set of connectors for several web services for genomic identification, and a powerful Silverlight visualization called SilverMap for analyzing these results. This video will highlight some of the benefits and behaviors of the assembly and alignment algorithms provided in the MBF library. I'm going to open a FASTA file that contains a series of protein sequences. Here on the left-hand side, tree view, you'll see the sequence information that was contained in the file. And on the right-hand side, the metadata and the associated protein information. Now you'll notice that some of the letters are highlighted, but most are not. By default, the Sequence Assembler is tuned for DNA analysis, but it also supports protein analysis. So I'm going to go in and I'm going to change my color scheme to use the standard protein color mapping. And there we get a mapping that's more appropriate for working with protein alphabets that contain color coding similar to other software packages used in alignment and assembly of proteins. So the next step then is I'm going to align these protein sequences using PAM-SAM. Now PAM-SAM is a multiple sequence alignment algorithm that developed in-house here at Microsoft Research, but based off the algorithms and the techniques used in the muscle tool that's widely available and used in the genomics industry. So we'll select PAM-SAM. I also need to change the similarity matrix from using ambiguous DNA to a PAM-70, which is used to align proteins. These two changes, I hit submit, and on a dual-core laptop, 3 GHz with 3 GB of memory and 64-bit Win 7 OS, we'll see how long this actually takes to align all these protein sequences on a pretty standard for today's hardware desktop. And here we go. So let me go to the report here. So we had 23 input sequences. We show that we used PAM-SAM as our alignment algorithm, and it took us just over 13 seconds to process these into a complete alignment. And if I go back to my alignment view, we can see how all these sequences align very nicely. I can zoom out and also expand the different sequences and query them and highlight them in the right-hand view to drill in further as to what I'm looking at. And I can also scroll up and down, and the two views, top and bottom, both stay synchronized in terms of what I'm looking at. And it becomes fairly obvious in the bottom view some of the mutations or differences that have occurred in this alignment, but nonetheless the algorithm and the input parameters that define the proximity requirements for alignment have also succeeded in finding a relatively good alignment for these sequences across the spectrum of the sequence length as input. Now BLAST can also be done on protein sequences, though the default parameters here must be changed to support proteins. So I'll have to change the program to BLAST-P. Now this is all specific to the NCBI BLAST program. The gap costs for proteins need to be a little bit higher, as well as the word size needs to be a bit smaller. And these parameters, if you'd use the default, the error messages would come back and show you the required changes to obtain a result. But those should be the only changes required, and I'll hit submit. And then we can see that the search is executing, and at this point we're dependent upon network traffic and the web service load at the time of execution. Really there's nothing happening local here, but to the researcher it's really just like a button click and part of the workflow process that they would use in any analysis. And here we go. This is a hit table as returned from a BLAST query that shows the different gene IDs and their relative closeness in similarity to the query sequence put in. Each of these can be evaluated and drilled into and examined, but there's also a SilverMap component as provided by the Queensland University of Technology, which gives a more visual representation in a heat map form of the same hit table. and uses the closeness in matching to generate closeness in visual proximity to the query sequence, which is always in the middle, 15373 here. So if I'm interested in maybe this guy who is very close in similarity, I can see this is a certain type of organism or gene. I can drill into others around the area, another ribosomal protein. If I wasn't sure what I was dealing with, and this is the way scientists would examine and identify the query sequence. And I can also change the center of query sequence to see how similar it is to all the other ones, and you can see that's a very similar to all the other returns. I can then go back to the original query, zoom in again, and I'm likely to get the same result on any of these because of the result from the first one. But again, I can go back to the query sequence and use that as my similarity matrix. So you can see variation between the query and the returns, but all the returns are highly similar. And this can be important in terms of how the research is performed. Again, I can go back to the summary. I can zoom, pan, decrease the size of either window, and perform all sorts of interesting research on DNA, RNA, or protein sequences. Thanks for taking the time to check out this video. To learn more about the Microsoft Biology Foundation, including more videos and download information, be sure to check out research.microsoft.com/bio. Thanks.